Welcome back, students. Okay, so now most of you joined. Yes, okay. Still people are coming, so we'll start. It's going to wait for 1 or 2 minutes, then we'll start. And meanwhile, we just can go through what we have done yet. And in our previous class, we talk about restriction modification system, that is the restriction and the nuclease. And in that we mentioned that there are two types of things. One is that endonuclease activity, second is the modification activity, and the modification activity that we are talking about is the methylation property. And in that we also discussed that restricted endonuclease are only cleave the enzymes which are not methylated. If the restriction site is methylated, restriction site methylated means the enzyme which is recognized in the recognition site. If the recombinant site is methylated, then it won't cleave. When you talk to the recognition site is a sequence on your DNA which you are actually recognize and produce a cut there. And what is the nature of it? We talk about it's around 4 to 6 base pair long, 6 to 8 base, 4 to 8 base pair long, and it is a palindromic in nature. Palindrome also mentioned two types are there. One is a mirror palindrome, and second is reverse palindrome. And then we talk about the different types of restriction nucleases, and we mentioned basically there are three, but now classes are keep on increasing 4, 5, 6 class, you know what I would say, but three are of much importance than others. And then we talk about type 1, type 1, type 1, type 2, 3, type, all of these we characterize on the basis of their properties, what type of action they do, and one of the four properties we are distinguishing them. One is their composition, second the product requirement, nature of the target sites, symmetric, asymmetric, and the position of the cleavage from where they are cleaving. These are three properties on which we are characterizing them. 1, 2, and 3. One we mentioned it was uh, cleaving at a remote site, that is around 1000 distance away. And then type 3 also is cutting at a remote site, but remoteness is a bit lesser, 20 to 30 degrees away. Then we talk about the type 2, and type 2 one is the which is living within the recognition site or just outside the recognition site. That is the property of the type 2. And we stopped at this slide, and this is the one from where we have moved today. So if any one of you have any query regarding that, I hope you have read the literature and read the lecture also, I think, watched the lecture again, I think. If you have any query till now, please let me know.
Sir, would it be fine if you repeat type 2 enzymes?
Sorry?
Sir, would it be fine if you repeat type 2 enzymes?
Repeat, I won't repeat again, just tell me the specific issue that you are having.
Sir, so it's you said that it'll be they cut within the recognition site or slightly away, right? So does that differ with
Just
the
outside, just outside.
Okay. Okay. Thank you, sir.
In detail, few more points are going to come in the future. So I don't think that we are completing type 2 now. And after type 2 to the power 5, in type 2 we will study in detail. Few more features are remaining. In the future slide they are going to come.
Okay,
Getting the
sir.
point? And simply that you are asking when I'm saying it's away so that. So this is the one. If you say that the sequence is done GTAATG, if you are cutting at a location 1, sorry, just a moment. Okay, so if you are cutting here, then you can say that it's within, and if it's cutting at this point, then this is outside. Getting the point?
Yes, sir, I understood.
Okay, so this is the only thing, whether either within or just outside, or we may say that just one nucleotide away, not like very far. Okay.
Okay, sir. Thank you.
This is the thing with your type 2. And type 4, it is not of much for you. You may skip it also, but it is good to know the whatever things are coming up. So this is a type 4, and this type 4 also, this is characterized on the basis of the 4 property that we mentioned. That how they are producing cut. Until now, what we are talking about type 1, type 2, type 3, we are saying restriction and modification system. And if the site is modified, that means methylated, then it won't produce a cut. But type 4 is the one which recognize the methylated sequences. It means if your recognition site is methylated, then it will produce the cut. So this is another. This is the property of this type 4. So the common feature of these enzymes that have the two enzymatic activity. One is the methyl transferase. So that means it can transfer the methyl group. So previously we were talking about that it was adding the methyl group, but it is the removing the methyl group. And the second property is the endonuclease. So this is the two property that both it is having, and both the properties are present on a single polypeptide chain. Previously, if you remember, in case of type 2, we mentioned that it have only your endonuclease activity, you don't have the methylation activity or methyl transferase activity, and another enzyme is there for that property. In case of type 1 and type 3, we mentioned that two different subunits are there. One subunit do the job of methylation, other subunit do the job of endonuclease activity, but here only single unit is there which is going to cause both the action, that is your methylation or methyl transferase activity or the endonuclease activity. So this is a property of your type 4 that is different. Then come to the cofactor requirements. So cofactor endonuclease activity is affected by SAM, but it is not necessary for it. The other means ATP, it don't need ATP. So whether you give the ATP or not give the ATP, there is no change in the activity of this type of endonuclease. And type 2 enzyme recognize modified, typically methylated, as I mentioned. So then that's a unique property. They recognize the methylated sequences, and it is very distinct from the other 1, 2, 3 of your some example. If you see here, R eco 57I, RBCE 83. So these are the few example of your type 4 restriction endonucleases. And type 4 restriction endonucleases are just say where you put them. So when we are talking about type 1 and type 2 and type 3, so type 2 we put it in. We said that type 3 is in between type 2 and type 1, because type 1 is producing cut at a very long distance. Type 3 is cutting near the very near, and type 2 within. So type 3 was within the type 2, and uh, type 3 is within the type 2 and type 1. So here if you are talking about the type 4 where it lies, so type 4 lies in between type 2 and type 3. So here the type 2S is written. So this type 2 is further subdivided into 3 groups. So that is not of much interest for you people. If you want to read about it, so you can read it, and if you have any issue, you want to understand this type further sub classification of type 2 restricted nuclease, you can ask me in extra something, but it is not for your mandatory for your course. So they lie there, and one more thing property is there for this type 4 restricted nuclease is that MR system is there, that is as we were talking about these restriction endonucleases, all the restriction endonucleases are the immune system of your bacteria. There is a protection from the bacteriophage infection. So all the role that we are talking about are because of the getting protected from the bacteriophage infection. So this was a bit few things about your type 4, and other than this type 4, there are type 5 is also there, and type 5 is the one that is the restriction enzyme, but we generally do not consider it as a restriction enzyme, but it is also a type of restriction endonucleases. So you all of might have heard about the CRISPR-Cas9 system. So this CRISPR-Cas9 system is at type 5. So here the recognition is not a type of the sequence that we are talking about in case of our type 2 and type 1, 2, 3, 4. Here the recognition is based on an RNA sequence. That RNA that we call the guide RNA. So wherever the sequence that's matching with the guide RNA, it is going to cleave it. So this is the property of this your type 5 enzyme. So that type 5 is also called as the CRISPR-Cas9 system. They can cut DNA of variable length. So whatever the sequence with your guide RNA is going to be there. So where this guide RNA is going to recognize, it is going to produce guide. And this Cas9 is an enzyme that is the endonuclease is the Cas9, and guide RNA is your recognition sequence. So whatever the sequence are present on this guide RNA, this Cas9 will recognize it and produce the cut. So this guide RNA have different motifs over it. One motif is for the recognition of Cas9, and the second motif is for the recognition of your DNA sequence. Okay. So there are different types of Cas enzymes are also there. Cas12 and Cas13s are also there. They are going to recognize RNA instead of DNA. So these are a bit another type of your restriction modification system. And in recently, previous year, Nobel Prize won for your Cas9 discovery, and there are the scientists who awarded for this. This is Jennifer Doudna and Emmanuel Charpentier. They are the two scientists who got the Nobel Prize for that. And some others than this type 1, 2, 3, 4, 5, we call that the modified enzymes or artificial restriction enzyme. Means you can design as of your choice. So till now, whatever the restriction enzyme you are talking about, so all these restriction enzymes were naturally isolated. Means they are derived from some bacteria. But when they are derived from some bacteria, it means they are going to have their specific recognition sequences, their specific locations. But as we are a genetic engineer or recombinant gene technology students, so we want our enzyme or restriction enzyme of our interest. Means we want this enzyme should recognize this sequence and it should produce a cut. So then you can engineer enzyme of your interest. So you can engineer an enzyme which recognize a site that you want and will produce cut there. So that type of enzyme we call it the artificial restriction enzyme or genetically engineered restriction enzymes or modified restriction enzymes, whatever you can call them. So they are engineered. So they are going to have two different domains. One is the DNA binding domain. DNA binding domain is a domain which is going to recognize the DNA sequence where it will go and bind. And second is going to be endonuclease region, which is going to produce cut into your DNA. So this is a bit few classes of your restriction endonucleases. So broadly, we are classifying under 6 class. So it is not just ending. So when the science keep on advancing, and so new and new restriction endonuclease systems are being discovered. Okay, so as I mentioned, 3 are of more interest. So here are a bit comparison between these 3. So comparison as first slide we are talking about, comparing all on the basis of where they are recognizing, what type of their composition is, what the structure is, what they require, what is the specificity for the host, previous site. So all these things are there in it. So if we talk about the retention and modification activity, so restriction and modification activity. So simple type 1 have the single modification activity. Type means both the things are present on the same enzyme. Type 2 have the separate multifunctional enzyme. Type 3 have the separate enzyme with the endonuclease subunit and the methylase. Protein structure, type 1 has 3 different subunits. Type 2 has only one simple enzyme, so no subunits are there. Type 3 is going to have 2 different subunits. Requirement, type 1 is going to need ATP, it needs magnesium ion, it needs SAM, where type 3 needs ATP, magnesium, and SN you don't need the where the type 2 it just needs magnesium ion, it neither need ATP nor need SN okay. Then the sequence for specific sites. So asymmetric thing is there in case of your type 3, so you can see that if P1 3 and P1 5, so there are different asymmetric is there, asymmetry is there, in the type 1 also. Type 2 rotational symmetry is there, so both the side, both the strand they are going to produce cut, so symmetrically they are producing cut. In this side, rotation. Type 1 is very remote, 1000 base pair away. Type 3 is 24 to 30 or 26 base pair away from the host specific site. Type 2 at or near the host specific site is the more specific one. Unlike turnover, there is no so turnover you can calculate for the type 2 and type 3. DNA translocation, you can translocate. Type 1 is helping in the translocation. What is helping the translocation because it is producing cut at very large locations, so translocation is there. Whereas type 2 and type 3 they have no in the DNA translocation. Site of methylation, host sites, host specificity site. So all the case site of methylation is the host specific. So wherever in the host, what type of enzyme you are getting from the host, depending upon that, it is going to produce the methylation. And here the few more differences are there. So it is going to be a modified type of as we are talking in the previous one. So similarly, this is some modified table. You may say that. So most of things are common. So similarly, you can get the one more thing that is the abundance. Type 2 is the most abundant, where type 3 is rare, and type 1 is less common than type 2. So this is the abundance. So now coming to the whatever the Maniswini was asking, how they are going to produce the cut, whatever they are, how you are going to deal with it, what type of a cut, and how it is going to. So as we mentioned that two things are there in your system. One is the recognition, and second is the restriction. And another property that we are talking about methylation. So methylation is not a property of type 2. So we are not going to talk about methylation here. So we just going to talk about the recognition and the restriction or the digestion. So two different domains are going to be there in your enzyme. One is going to be recognition, second is going to be digestion. So first simple thing, as you can see, just a moment. So as you can say that this is the DNA. DNA is going to be double stranded, and in this DNA there is going to be a recognition site, and where in this recognition site your action randomly is going to produce a cut. So first simple thing it have to recognize. So where but it will go. So first thing there is a non-specific recognition you can say. So non-specific recognition means it have to recognize DNA. So there is a DNA binding domain which is going to recognize just DNA. So randomly wherever in the DNA it is going to, it is going to be non-specific. So once it bound to DNA, now it is going to have to search for its site where it have to produce a cut. So that is called the specific binding. So once it's bound to a non-specifically, then it glide over your DNA sequence. Like you can see, it's non-specific binding here, but it is not going to produce cut here. So what happened? It may leave this space also. So because recognition sequence is not this one. So then what it do? It glide over it. Move over, it scan your DNA. While it's scanning, it search whether the sequence of interest is there or not. If your restriction endonuclease doesn't find a sequence of interest, then it will go off. But when it found its structure of sequence, then it invade within it. When it invade within it, what it do? It is going to break. It has the helicase activity. Helicase activity means it is going to break the helix. It is going to break the hydrogen bond. Then it have the nuclease activity. So nuclease activity means it is going to break the phosphodiester bond. So then it produce a cut here and it produce a cut there. So to cut it produce, it is going to produce type 2 resting nucleases are there. So all the resting nucleases are going to produce cut on the both the strand. So they are going to produce cut on both the strands. So they release two fragments, one fragment and this fragment. Okay, so this is the way how your restriction element nucleus type 2 they are going to act. Okay, so is it clear to you? Is it clear how the type 2 is acting?
Yes, sir, it's clear.
Okay, so now when they act, what type of cut they produce? So depending upon their cutting habit, they produce two type of a cut. One is the blunt and second is the sticky. So if they are producing cut at the same location on the both the strand. Okay, so for example, this is the sequence. You can see the upper sequence in the sequence A. So you can see the upper sequence A. So here the sequence is A, G, C, T. So here L1 is the name of enzyme. So this L1 is recognizing A, G, C, T. So here the how many sequences it is recognizing? It is recognizing 4. So it means this L1 is a tetra cutter. It is going to cut. It is going to recognize a 4 sequence. So when it recognizes these 4 sequence, it produces cut at GC in between G and C. So it produces cut. It produces a phosphate has to go between the G and C. So both the stand at the same location. So when it is cutting a both the stand at the same location, it is going to produce a blunt. Means no overhanging thing is there. So such type of cuts, such type of ends we call the blunt ends. So nothing is pointed out, nothing is hanging over it. So these are called the blunt ends, or also called the blunt cutters. So those enzymes which produce blunt cut, means they are cutting at the same location on the both the strands. So such type of enzymes are called the blunt end cutters or the uh, yes, it's a blunt end cutters. Okay. So another way of the cutting is a sticky end. We produce a sticky. So why are the term sticky is there? That I will tell you when I'll talk about it. Okay. So second thing is the sticky one or the staggered cut. So blunt cut or staggered cut. Staggered cut means they are not cutting at the same location. They are cutting at a different location. So different location means if, as in this previous case, it is producing at G and G sequence. So in this example, so like this is a sequence GAATTC. So name of the enzyme is EcoR1. EcoR1 recognize GAATTC. So one possibility is there to produce at same location. So for example, it produce cut here. So it produce cut here in the both the stand. It is going to produce a blunt end. But as I mentioned, it is not producing at the same location. It is producing different location. So it may produce cut here, then also it may produce cut here, it may produce cut here, it may produce cut here. But EcoR1 recognize the sequence and produce cut between the G and A. So G and A. So as I mentioned, the property of the recognition sequence is the palindromic nature. So the sequence when they are palindromic in nature, then it produces cut only at the palindromic sites. Means if found G and A, then it is going to produce cut in between G and A. Otherwise, it can't produce cut. Okay. So here it is producing cut here, then it's producing cut. What is going to happen after this cut? So you can see. So if so when you separate these two, if one cut is produced here, another cut is produced here. So what is going to happen? This region in the both the strands AATTCC. So there is a hydrogen bond and they are held together by the hydrogen bond. So what, as I mentioned, the restriction ligands are also breaking their hydrogen bonds. Okay, so that's why the strands are what separated. But in the absence of any enzymes, when the no restriction property is there, if you have such type of things, means when you cut it, what type of the you are going to get? You're going to get G in the one strand, in the second strand, you're going to get as C, T, T, A, A. Okay, so these are the hanging regions are there. So these hanging regions, these are pointed regions are there. So that's why this end is not a blunt one. This end is a sticky one. So why it is a sticky one? So as we all know that nitrogenous base has the property of making hydrogen bond. So and this the formation of hydrogen bond is on the basis of complementarity. So any single stranded sequence which has a complementary region to it may pair with it. So this property of pairing of the single stranded DNA makes it a sticky image. Okay. So this end is called a sticky end because it can pair with a complementary sequence. So what complementary sequence it can pair? It can with anyone who is going to have as AA and TT. So any strand which is going to have a AA and TT overhanging region, it can pair with it. So such type of sequence are called, such type of cutting are called sticky end production or sticky cuts, or you can call it as a staggered cut. Okay. So L1 was an enzyme which produces the blunt end, E carbon which is recognizing GAATTC. It produces a staggered cut, and staggered cut leads to the production of the sticky ends. Okay. Getting this point? Getting it?
So can you repeat once again?
What should I repeat?
The last part.
In the last part.
So staggered one.
Staggered means it is producing cut at different location. In the one strand at one location, in the second strand at another location. That is called the staggered cut. When they are producing a staggered cut, then like cut is like this, in this way. So a cut is like this. On the one strand it is cutting at this location, the second it is cutting at this location. That leads the production of an overhang in the both the strands. So overhang means it is going to have one strand TTAA, another strand is also going to have TTAA. So these overhang having a single stranded DNA sequence, that's why we are calling them the sticky ends, because single stranded DNA have the ability to pair with any other strand. So another example of similar type of enzyme which are producing staggered cut is BAM-H1. So what BAM-H1 is doing? BAM-H1 is recognizing GATC, GATC. So GATC is a 4-nucleotide sequence, means BAM-H1 is a tetra cutter. It is going to produce, it is going to recognizing 4 sequence. EcoR1 recognizing GAATTC means 6 sequence. It is a hexa cutter. Okay. So another example is Beagle1, Beagle2, BEGL2. It is recognizing again the GATC, means the recognition sequence of both Beagle BAM-H1 and Beagle2 GATC. Okay. Another example is South3A. South3A is also recognizing 4 sequencing. G, it is also recognized GATC. Okay. So these are the 3 different enzymes. All the enzymes, BAM-H1, Beagle2, and South3A, they are recognizing for they're recognizing sequence, and when they're recognizing sequence, they are going to produce a 4 nucleotide overhang, and these 4 nucleotide overhang after producing a cut, they are going to have a GATC overhang. Okay, what the point?
Yes.
Okay, so one thing is that the two types of the cut your enzyme is going to produce. One is going to produce a blunt end cut. Blunt end cut means it is going to cutting at the same location. So when it is cut here at this location or this location, any location, any sequence, forget the name of the enzyme, forget the recognition sequences. Okay, so just remember that blunt means it is going to produce cut in the both the strand at the same location. That is going to be a blunt end cut. Second is a staggered cut, means they are not going to produce cut at the same point, they are going to produce a cut at a different point. Like in case of this one, like if this sequence is there, GATT, forget this echo R1. So if you produce cut at G at location 1, if you produce cut at this location, then the hanging part is going to be this one, and then I'm not writing, so G is going to be here, and C, then T, 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 A, okay, and here also it's going to cut at here, so then the reverse is going to be this one. In the second case, you may say that it may be cut here, then what is going to occur? Then G, A is going to be here, and C, T, T, A. So this overhanging is going to be these 3 nucleotide sequences. If you are saying that it is going to produce cut here, then it's going overhanging is going to be so on. Okay. So that overhanging depends upon where it is going to produce cut. So as I mentioned, enzymes are producing symmetric cut. So the symmetry is going to be in a way that both the location, both the strands, cut is going to be symmetric. It is not going to be like that. In one strand is going to produce cut at this location, another one is going to get this location. So as you are moving in this direction of this strand, the second strand also you have to move in the opposite direction. So at G, here also it is going to be G. A, here also it is going to be A. A, here also going to be A. So if you are producing cut at A, then the ends are going to be blunt end. Okay. Is it fine?
Yes.
Yes, sir, it's fine.
Okay, so in the lower one, the C panel, we have there are 3 different enzymes. So what we are talking about the sticky ends. So there are different enzymes. They may recognize different sequence, and they may produce a same overhangs. So these are the these are we are calling them as a overhang because they are hanging. They are hanging over to the double-stranded sequence. So these to them we call them overhangs. So different enzymes may produce the same overhangs or same sticky ends. So if we take the example of first one, BAM H1. So the recognition sequence of BAM H1 is this one. Okay, G, G, A. So sequence that is BAM H1 is G, G, A, T, C, C, C. Okay, so here is going to be G, G, A, T, C, C. So both are reading same. So that's why they are the palindromic in nature. Bigger one also recognizing this sequence. Okay, so the growth sequence of bigger one is going to be A, G, A, T, C, T. So positive pair of right, A, G, A, T, C, and T. Okay, AGAT, TCT, AGA, TCT. So this is also a palindrome sequence. Okay, so both the enzymes The recognition sequence of the both the enzyme is going to be 6 sequence, hexacutter, and the cut is within the sequence. Where is being cut? Within the recognition sequence. Now, another example is going to be the South 3A. South 3A recognition sequence is going to be G A T C. Okay, so the is going to be G A T C. So recognition sequence is 4 nucleotide sequence, but where it is position card? It is in position card outside the recognition sequence. Okay, in the both one within the recognition sequence, in this one just outside the recognition sequence. Okay, what the thing? What is the how within the or what do you mean the outside? Got the point or not?
Yes, sir. Yes, sir. I understood.
Yes, sir.
Okay, so this was about the blunt and sticky ends. Okay, so this is again the similar representation of sticky ends and the blunt ends, how they are going to produce the cuts, different enzymes. Here also HIND3 is another example of enzymes that is going to produce sticky ends. And the recognition sequence for it, this is the AAGCTT. So this is going to produce cut between the A and the AA, so symmetric cut. So both the symmetrically it is going to produce cut between the A and A. Within the sequence it is going to produce cut, and the ends are going to have overhangs. So overhangs is going to have T, C, G, A. So or you can say like 5 to 3 prime A, G, C, T, A, G, C, T. Okay. So another example is SMA1, SMA1. So it is going to produce a cut blunt end. So blunt end it is going to produce cut between C and G. C and G. Symmetric cut is there. 3 nucleotide this side, 3 nucleotide this side. So we are going to get a blunt end. EcoR1 we already have studied. The EcoR1 is producing symmetric cut between the G and A. G and A symmetrically they are producing cut. So symmetrically is going to be sticky end. Dimetch1 we already studied in the previous one. It is also going to produce a symmetric. So GG and GG. We are going to have overhang of GATC. Okay. So in this way, your restriction enzymes are going to produce cut. So here are some list of enzymes that you have to remember, because for your exam purpose or for your study purpose also, you have to give some example of your enzymes, what their recognition sequence is, and what type of cut they are going to produce. So here are the few important enzymes. So in future also, if you are going to work in your laboratory, so these are some important enzymes that you have to use. So ECOR1 is enzyme you got from the E. coli, Escherichia coli, GAATTC, the sticky ends, Dimetch1, Bacillus aminoglycations, GGATCC, Beagle2, P1, PBU2, HIND3, HIND1, South3A, L1, DAC1, Thermophilus aquaticus, HAE3, Haemophilus aegypticus, NOT1, Nocardia, otitis, Caviarum, SF51, Streptomyces, Fimbriatus. So these are the things which are going to recognize it. So here you can see in the last one, it is going to have a NN in between. Those wherever this N is written, this N means anything can be there. Here any random sequence may be there, but for the left and right side of the sequence, GGCC, GGCC, if it is there, it will recognize, otherwise it won't recognize. So recognition is for GGCC and GGCC. So this GCC may be present from the 1, 2, 3, 4, 5. 5 random nucleotide may be there, but 5 random nucleotide may be surrounded by GGCC. Then SF51 is going to recognize it, and it is going to produce a sticky. Okay. So these were the some property of it. So now came to the how you have to name it. So nomenclature of restriction enzyme is also an important thing. So if you can see here itself, so the name in the restriction enzyme is going to be derived from their source organism. So as you are saying EcoR1, so EcoR1 is also the E. coli. So this is the nomenclature how you are going to name it. So, so Smith and Nathan in 1973 they give as they recognize it, and they give the nomenclature, and they said that you should name restriction enzyme depending on their source of the origin, followed by their strains and the number of enzymes isolated from that organism. So simple one. So the first is there's the E. coli is there. Actually, this is E. coli and O. So eco is the first three, then R is the strain name here, and the 1 is the first enzyme isolated from this E. coli. Then eco R2 is going to be there. This is going to be the second enzyme, which is going to be isolated from the Escherichia coli R strain. Then the eco R3, if it is there, then it is going to be the third enzyme, which is going to be isolated from the E. coli. So depending if the strain and the species are common, then the first four letters is going to be remain same, then followed by the your number of the your enzyme. So the every enzyme must contain three-letter code followed by strain and number. The first letter is going to be there from the first alphabet of the source, that is the genus. Second, third letter will be from the species, and the fourth letter represents the strain, and the number represents the number of enzymes produced by the strain. So whatever the strain that you are talking about, like if you talk about any of the student that we select, example the Adish Mishra, if the Adish Mishra is Adish is his genus name, Mishra is going to be the species name, then the enzyme isolate from the Adish, it is going to be AMI. So A from the Adish, MI from the Mishra. And if the individual one, no strain is there, then the strain is not going to be there, then if one enzyme is isolated, then that enzyme we call it the ADMI1. Similarly, Atoiba, if you call it the Atoiba Thokchom, then name of the enzyme will be ATH1. The enzyme is the first. So, as in case of the restricted endonuclease, they are isolated from the bacteria. So, depending on their species, and bacteria also have the strains, and you name accordingly. Okay. So, after the nomenclature, another important property of this enzyme is called the star activity. So as we have discussed here that every enzyme recognize a specific sequence. So as we all know that they are the part of the immune system, so they recognize specifically. So as in case of human also, in case of immune system, you call them the cross activity. So cross activity means if they recognize some non-specific thing. So that non-specificity is also there in case of your restricted enzyme also. So how this cross-reactivity or a non-specificity arises in it? So this arises on the basis of what condition you provide, what buffer conditions are there, whether the every condition that you think that you're talking about is appropriate or not. If any condition changes, then we may say that the star activity may be there. Star means another location than your enzyme specific location. Star activity is the relaxation or alteration of the specificity of restricted enzyme mediated by the cleavage of DNA. That can occur under reaction condition that differ significantly from those of the optimal condition. So as you mentioned that it required magnesium chloride, it required magnesium iodide, it required a pH. So if you change the pH or if you change the buffer condition, just a moment, student. Okay, students, welcome back. Okay, so if any condition you change, any optimal condition you change, then specificity is going to change. So this change in the specificity we call the term star activity. So star activity is used by the layer who characterize the modified activity in eco-R1. So you are studying eco-R1 and different optimal condition, and we found that not just specifically it is going to be some non-canonical sites also. That is called the loss of specificity or the loss of specificity or something called the star activity. So when you any enzyme you got, you must check the star activity present in that enzyme. So it means that enzyme may possibly, if you change some upper conditions or some dilution, if you are changing, that leads to the non-specific activity in your experiment. So a star activity can happen because of the dilution line as seen in green. So there are some other more different examples are there, different types of the enzymes, different things are there. So that leads to the star activity. Okay. So another thing, one more property of your enzyme is restriction enzyme that we are talking about. It's called the isocytomer. So as you know that the isomers are there. So isomer that you are talking about, these isomers are those enzymes which are having same substrate activity. So similar type of the isomeric property we are talking about here also. Iso means same. Isomers means they are producing size of the cuts that we are talking about. That you call the isomers. So what are the definition wise? Definition wise is a pair of reaction enzymes specific to same recognition sequence. So when we so what type of the enzymes are going to be? So they are the enzymes which are you got the enzyme from two different sources. So if you get an enzyme like from the E. coli, any enzyme you get from the E. coli which is going to recognize same recognition, then it is not going to be isomer, then it is going to be a same enzyme. But if you isolate an enzyme from a different organism, like in case of SPH1, Staphylococcus is going to be the name of the genus from which this enzyme is going to be isolated, effectively the BDU. So Bacillus is going to be the genus of the enzyme from which we are going to get. So both of the enzymes they are going to follow the same sequence C, G, E, A, C. C, G, E, A, C. And going to produce cut between C and G. C and G. So such of the type of the enzymes are called the isocytomer. Okay. So the first enzyme discovered which recognized the hidden sequence is known as the prototype. All subsequently also identified as enzymes that recognize the sequence for as the isosomers. So these are the isosomers. So one more type of the isosomers are there, means they recognize the same sequence. They are recognizing same sequence, but producing cut at a different location. So first example in which we have taken. So in this case, this enzyme and this enzyme both are producing cut at the same location, that is this one and this one. So then we call the isocytomer. Another one, the enzyme that will be the same sequence, that means sequence is going to be the same, but the location in which they are going to produce cut, they are going to be different. So the example here is the SAMA1 and EXAME1. So the enzyme isolated from the two different organisms, they are recognizing CCC, GGG. The sequence producing here also CCC, GGG, but SAMA1 is going to produce cut here, simply means it is going to produce a blunt cut, whereas ZAMA1 is going to produce cut at this location, it means it is going to produce a sticky end. So this type of enzyme we call them the neosizomers, recognizing the same sequence but producing cut differently. Another example is KPN1 and ACC 651. So it is going to produce cut this location and here at this location. So these are the enzymes which are producing cut at different location in the same recognition sequence. Such type of enzyme we are calling them the neosizomers. Means new size you are producing. So they are iso size, same size they are producing, neo size and new size they are producing. Okay. So one more type of the enzyme is there, that the isomerism is there in it. An enzyme that recognizes slightly different sequence. So what I think they are recognizing different sequence but producing same ends. So in previous slide, if you remember, yes, here in this one, BAMH1, BIGL2, and SOU3A, all they are recognizing different sequence, but what type of ends they are producing? They are producing same type of ends. All the are going to have GATC overhang. So such enzymes which are recognizing different sequence, but producing same overhang or same hanging parts, that types of the enzymes we call them the isopodomer. Isocytomer, same recognition sequence, same uh, restriction sites, same type of overhang, everything is same. Neosizomers, same recognition sequence, but after the cut is going to be different. End product overhanging part is going to be different. Maybe blunt ended, maybe sticky ended, so different restriction sites are there. And third type, different recognition sequence, but same overhangs are going to be there. So these are three types of the isomeric form unit in your enzymes. All the enzymes you are going to get from different source. Enzymes that are produced by different organisms that produce, but at different same or the different overhanging way. So these are the types of the enzymes. Now come to their utility. Why we are studying them? We call these restriction enzyme as a molecular scissor because as Taylor need his scissor for tailoring his items. So these are also your molecular scissors. When you want to tailor your DNA, you want to make your DNA to work in your way. You need these enzymes. In detail, what type of activity they are going to do, we will study in our next class. Now we are ending to our session, so I'm stopping now. And before ending, if any one of you have any query till now what we have discussed, please ask. Any query?
No, sir. But can you just share this PPT with us?
Yes, I will share you.
Yes, sir, please.
I will share you this PPT on your MNS. So don't worry.
Okay, sir. Thank you, sir.
Okay, so as this module will be done, I will share you all the slides of the module. Okay, or I will share you even before the ending of this module. Just today itself, I will share you. Any other query other than this? Nothing? Okay, so then please turn on your camera while I call your name. Just a moment. Demetrius,
Present, sir.
Tridev, Tridev,
Present, sir.
Manaswini,
Present, sir.
Adish,
Present, sir.
Yuvraj,
Present, sir.
Sanjana,
Present, sir.
Nehal,
Present, sir.
Atoiba,
Present, sir.
Devanshi, Devanshi, Nandini, Nandini,
Present, sir. Present, sir.
Kshitij,
Excuse me, sir.
yes, yes, Devanshi, you have been marked absent.
Sir, I'm present. I'm having network issue.
Janvi,
Present, sir.
Shivani,
Present, sir.
Shivani, Shakti,
Present. Present, sir.
Rupal,
Present, sir.
Sahiba,
Present, sir.
Ishita,
Present, sir.
Amrita,
Present, sir.
Tanvi,
Present, sir.
Yukta, Yukta,
Present.
Yukta, your camera is not on. Ayushi,
Present, sir.
Vivek,
Present, sir.
Vivek.
Present, sir.
Okay, so thank you all. Stay safe, stay happy.
Thank you, sir. So, Saiba this side, was I audible because I turned on my camera also?
All are present.
Okay.
All of you are present. Okay.
Thank you, sir.
Thank you, sir.